Okay, I think that I covered the first few objectives of section 2.2 more extensively in the previous one than maybe I needed to. So I'm going to kind of just use this time in this video to go through the last 6 standards that are going to focus more on energy capturing and harvesting and how energy is produced. And this is equivalent to book section 4.4 through 4.5. And I think I had some animations, but I'm probably just going to talk through it just to kind of make it go maybe a little bit quicker. Um, all right, let's talk about the chloroplast first. And I like these 2 pictures. These are just like kind of 3 looks at the same thing. But in the chloroplast, everybody knows the chloroplast is used for photosynthesis, but there's a couple different things that are going on in the chloroplast for this light harvesting and eventual production of sugars that's happening. So on these stacks of membranes, the membranes themselves are called thylakoid membranes, Thylakoid, thylakoid membranes. And then a stack of thylakoid membranes is called a granum. The total of them they're called grana for plural. And the light is harvested through these membranes, and it's done through a series of different proteins that are in integral membrane proteins that are in the membrane of the thylakoids. And I'm going to probably say this more than once, but next unit we're going to go through all the details here. So for now, we're going to just do an overview. So for now, don't worry about every single detail, just the things that I'm stressing here. So energy from light is harvested on those thylakoid membranes, and then that energy is used. That's called the light dependent reaction because, of course, sunlight is required to be in that reaction. Oxygen is a byproduct of that reaction. And then in the stroma, or the fluid-filled portion of the chloroplast, are called, they used to call them the dark reactions, but they don't happen, they'll happen in the light too. They just don't require light to happen. So it's better to say light independent reactions, and that's also known as the Calvin cycle. So in that cycle, carbon dioxide is going to be synthesized. Is going to be used to synthesize sugars. Not the carbon dioxide isn't synthesized. It's going to be used to synthesize sugar using the energy that was harvested from the light dependent reactions. So that is, in a nutshell, photosynthesis. But let me just go through it again. I did that pretty fast, so let me go through one more time. The light is harvested on the thylakoid membranes. Oxygen is produced there from water as a byproduct and is given off. And then the energy that's made from that is going to drive a series of reactions that happen in the fluid-filled portion, or the stroma, where carbon dioxide is going to be taken in and be synthesized into larger molecules that we call, or that are sugars or carbohydrates. Okay, and then this just kind of shows you what an electron micrograph of the chloroplast looks like. You can see those stacks of thylakoids, or the grana, and then the parts that are in between there are the stroma. So the light dependent reactions, again, I don't want you to memorize so much. Like, just think of, those are those little poker chips. They're going to use to be harvest light energy. That to me is intuitive, that those are going to be, they look like little solar panels, that's what's going to be used to harvest the light. And then the stroma is going to be the part where that energy is going to then be used to bring together CO2 molecules to eventually build a molecule that's made up of larger numbers of carbons, like glucose, C6H12O6. And then that's just kind of like a cartoon look at it. The next organelle that we're going to talk about is the mitochondria, and these are not opposite of each other. It's not like animals have mitochondria and plants have chloroplasts. Plants have mitochondria as well. They're going to take those sugars that they use that are made in the chloroplast, and then going to use them for energy in cellular respiration, just like an animal does. Okay, so this is a kind of same look at it where you see the folds of the cristae and the inner membrane through the transmission electron microscope, and then that's kind of the cartoony version of it. This picture here is kind of a basic picture. I probably showed another one in the previous video, but so you have an outer membrane and you have an inner membrane, and the inner membrane has the enzymes that are going to be used for accepting electrons and then eventually be used to make ATP, and those are called ATP synthases. And then the matrix of the mitochondria is where the Krebs cycle is used, and the Krebs cycle is going to produce molecules that are going to then be used by the electron transport chain on the inner membrane that contains the enzyme ATP synthases, and that's how lots of ATP can be produced when oxygen is present in the mitochondria. All right, and then this is just kind of, again, another overview of it. Next chapter, next unit, we're going to go through in much, much more detail. But essentially in the cytoplasm, which is not necessarily labeled here, but you have glucose, and glucose is broken down through a process called glycolysis. Glycolysis will happen if you have mitochondria or not, or if you have oxygen or not. It's not going to produce very much energy, only 2 ATP, but it's going to produce these 2 molecules of pyruvate. And when you have oxygen, when you have mitochondria present, those pyruvate can be shuttled into the mitochondria, and they're going to go through something called in the see, this should be cytosol here. I don't like this diagram, but at any rate, in the Krebs cycle, they are going to produce molecules, just again a little bit of ATP, and produce molecules that are used to carry electrons and go through this series of steps called an electron transport chain, where eventually this ATP synthase protein in the folds of the membrane are going to produce lots of ATP. So depending on how the pathways it takes, it could produce anywhere from 32 to 34, and altogether then the whole process is producing around 36 ATP. Okay, so 36 to 38 ATP. And this is just kind of the same look at it. Essentially, we're going to take in oxygen, glucose, and we're going to break it down into CO2 and water and energy. Just another very quick overview, but the process of glycolysis happens in the cytoplasm, and it's going to produce a molecule from glucose. It'll produce 2 molecules of pyruvate, and then those are going to be shuttled into the mitochondria, where a small amount of energy and these things called electron carriers are going to be produced by the Krebs cycle. Also, that's the point in which CO2 is going to be given off as a byproduct. And then eventually those carriers are going to go through something called electron transport chain and produce lots of ATP. And that happens in those different parts of the mitochondrion. Okay, so that was just a very quick overview of both the chloroplast and the mitochondrion for the purposes of just understanding kind of the very basics of how energy is harvested and produced in chloroplast and mitochondria.